Hey, this is Mr. Herzon, and today I'm going to talk about DNA structure and replication. Starting off with every living organism has four different types of organic molecules. And you remember this from our biochem unit. There's carbohydrates, lipids, proteins, and nucleic acids. Okay? Also, should remember that nucleic acids are made of a monomer called nucleotides. Okay? And there are three components to the nucleotides. There is the sugar, the phosphate, and the nitrogenous base, or you could simply say um, nitrogen base, but I'm going to spell out nitrogenous base. Okay. There are two different types of nucleotides, and since I'm going to be drawing um, both parts, um, I'm going to draw um, each each one. Okay. Now each one has a phosphate group, and the phosphate group has a formula of PO4, and double bond oxygen there. Okay. All right. And also, um, there's one over here for this second type. Okay. Next is our our sugar molecule, and here is one of the differences between these two different types of nucleotides. So, to this oxygen is bonded a carbon molecule that has two hydrogens. Okay, so let's put them both there. All right. Now each of these will be attached to a ring structure that's in the shape of a pentagon, and there is carbons at each one of these. So let's draw this one over here. Oxygen. We'll make the carbons inside of the pentagon more. Okay. Now, molecular biologists, biochemists, what they do is they count each of these carbons. So starting off at, oh, gotta get that into view. Okay, there we go. All right. Starting off with this first carbon molecule, it's called 1 prime, and then 2 prime, 3 prime, 4 prime, And then this carbon is the 5 prime. And let's do the same thing with this nucleotide. And the 5th one. Okay? Now, attached to both the second carbon atom is an OH group, as well as to the third carbon atom has an OH group. Now, what's different in this nucleotide, there is an OH group coming off of 3 prime carbon atom, but on here, the oxygen has been lost, right? So this is one of the big differences between these two, and we can circle it here. Okay. All right. Now the only thing that uh, needs to be added is our nitrogenous base, and I'm going to go ahead and draw the chemical structure of adenine for both of these. Of course, it could be any one of the 4 nitrogenous bases. Okay, and we'll get to those in just a minute. All right, so the nitrogenous base is connected to this first carbon atom. Okay, and the nitrogen right there, and this will form a ring structure. Okay, getting tight on space a little bit. All right, there is an NH2 group up here. There's another nitrogen in the ring right there. Okay, and still a couple more nitrogens. Okay, and there are double bonds located within the ring. Right here. Okay, and then one right here. All right. Now this is the nitrogenous base, adenine. In this type of nucleotide, there also could be uracil, cytosine, and guanine. Okay, it's this uracil that is a big difference between these nucleotides. All right. Now, um, this nucleotide here, oh, this sugar molecule here, is known as a ribose sugar. And that is another difference between the two. This nucleotide that's over here, okay, since it's lost its oxygen, it's deoxyribose sugar. And let's go ahead and add that adenine. NH2. Okay, we'll put in our double bonds. Okay, and then we're all set. Okay, so these nitrogenous bases, this one is adenine, but instead of uracil, this one has thymine, and it also has cytosine. Let's get that in the picture here. Okay, and guanine. Okay, so there's quite a few differences between these two nucleotides. This particular nucleotide is called the RNA nucleotide. Okay, this one over here is called the DNA nucleotide. Okay, now um, for RNA nucleotides, it's the building block for an RNA molecule. Okay, um, there there are some RNAs, all right, that are double stranded. Not very many. I'm running out of it. Already ran out of ink on my black Sharpie. Okay, so double stranded, all right, and and it's a few viruses have double stranded RNA. Okay, one of the more notable ones is the rotavirus, which causes um, it's very contagious, and it causes really, really terrible diarrhea in infants and young children. And it's responsible for over 200,000 deaths of young children every single year. Okay. Now, most RNAs, all right, are single stranded. Okay. And yes, there are some viruses that are single stranded. Most note that have single stranded RNA. Yellow fever is one type. Okay. West Nile, Ebola, all right. And not to forget coronavirus. Okay. And this would include SARS, MERS, and COVID 19. Okay. Now, most of the RNA molecules that do exist that are single stranded are in plants, animals, fungi, and humans. Going to give them a different distinction. Okay. There are three different types of RNAs. All right. And we'll explain these a little bit more when we get into protein synthesis. There is ribosomal RNA. Okay. The chief function of ribosomal RNA is this is the site of protein synthesis in the cytoplasm of the cell. Right? There is transfer RNA or tRNA. And the job that this has is transferring the amino acid to the ribosomes. And then the last type is messenger RNA. And this carries the gene sequence from the nucleus out to the cytoplasm and to um, to the ribosomes in the cytoplasm to begin protein synthesis. Okay. All right. Let's go. You can flip over um, your worksheet, or it's just like go to the next page. Okay. Now, for this, you can. I'm going to switch to a darker color. Okay. Now, for a lot of these drawings, okay, you can refer to the bio preview entitled DNA Structure and Replication. Okay, so the structure of DNA, it's a double helix. So, in drawing this, I'm going to make one of these strands here. All right, just like that. And okay, now um, here, well, let's not do that just yet. Let's go ahead and make our second strand. Okay. All right. Um, so this is the DNA structure, or the structure of DNA. All right. Let's label our strands. We're going to note that this is the 5 prime. And remember that fifth carbon atom, okay? That was numbered, um, 5 prime. Uh, um, uh, in deoxyribose sugar and also ribose, okay, as well. All right. This is going to have not one, not two, but 3 phosphates attached to it. All right. And then let's follow this down to the 3 prime end. Remember, it has an OH group uh, at the very end. Okay. Correspondingly, this other strand here is in the opposite direction, and that's because DNA is anti-parallel. Okay, so this is the 5 prime end down here, and there would be 3 phosphates. Okay, all right, and as we come back up here, this up here would be the 3' end. So let's put our 3' in the OH group here. Okay. Now, in terms of base pairing, all right, I'm going to use just a particular shape um, to denote um, my nitrogenous bases. Okay, I'm going to use red for guanine, and I'm going to use orange for cytosine. Okay. Now, there will also would be a sugar molecule located right here on both sides. All right. So let's go ahead and um, name our sequence. Okay. So here is adenine, and that only pairs with thymine. Let's use that in blue. Okay. And let's continue. Let's do thymine over here. And with adenine. Okay. And why don't we switch? Have an adenine here. And then paired with a thymine. Okay. And then we'll go ahead and have cytosine. And we'll do another one down here. And these are going to pair with guanine. Okay. Now, in terms of the backbone of DNA, we have um, the backbone is made of sugars and phosphates. We'll continue this all the way down. And there would be a sugar here, but I'm not going to go ahead. It would be hard to make this rung of, I guess we can color it in. Let's see, there would be a guanine here. And let's try having cytosine like here. That really doesn't come out too well, all right? But let's go ahead and continue to label our phosphates and our sugars. Okay, and I'm going to go ahead and put one more match down here. I have a thymine, okay, with an adenine. Okay, and do the same thing on this side. Phosphates, sugars, okay, sugars and phosphates. Okay, all right. Now the only other thing that we have to put in is our hydrogen bonds. Okay, between guanine and cytosine, there are three three hydrogen bonds. Okay, all right. And with between adenine and thymine, there's only two. Okay, all right. Now, one complete turn of the DNA molecule. So if we started here, all right, here's a half a turn. Here, we're back again at the original position. Okay, so from here to up here is 3.4 nanometers. Okay, all right. And every complete turn, I'm going to fall short. There should be 10 base pairs. And I think I only have six. 1, 2, 3, 4, 5, 6. Oh, I got a 7 here. I do have some space to put that in. But every 10 bases, that is one complete turn. Okay? And we should label our hydrogen bonds. Okay. All right. So this shows you the double helix. That's why I wanted to show this to you. And notice the opposing strands are in opposite direction. So we have a 5 to 3 direction going this way, and the 5 to 3 is anti-parallel. You can see it a little bit better on the model on your guided notes. Um, I can do the same thing on this side. I'm not going to go ahead and draw as much um, as what's in the drawing or the picture, the image that you have on your guided notes. All right, but let's go ahead and, oh, the black is back. Okay, that's good to know. All right, so I have that carbon atom right up here. And let's do another one here, another one here. Now, I said I wasn't going to draw that many, and what am I doing? I'm drawing all of them. Okay. Drawing really helps me with, over the years, with understanding the complexity of molecules, and especially everything in that's affiliated with biology. Okay. And I also taught molecular biology for 5 years. So I really got to know the construct of DNA and RNA and proteins over the years. Okay, so to this, we'll have um, three phosphates. Oh, we need a new color. So this is going to be our 5' N, and we would have a single phosphate that is going to be bonded to two of the sugar molecules. And the location of that is going to be at the 3' carbon. So I want to draw all in here. Okay, 2', 3', 4', and here's our fifth carbon atom, and one more set. Okay. All All right. So the bonds are between the fifth carbon atom and the phosphate, and the 3 prime, um, um, the 3 prime of the next sugar molecule, and that would continue up here. Okay. Now these are called phosphodiester bonds. All right. Okay. So here's our 3 prime end right here. And we would have an OH group there to signifying them. Okay. So you'll notice that DNA synthesis is occurring in a 5 prime to a 3 prime direction. Okay. All right. Now let's go ahead and add our nitrogenous bases, and those come off of the 1 carbon atom. All right. And I'm going to make this one guanine. Okay. Um, should we? No, we won't have another guanine. Okay. Um, we'll have adenine. Okay. And then cytosine. Okay. And let's see. How about thymine? Okay. So there is this strand. Okay. Now, to make the complementary strand, I'm going to have to either write this upside down, or what I like doing with the paper is just flipping the paper over. Okay. And now I could make my opposing strand that would be in the 5 prime to 3 prime position. So let's go ahead and draw our 5 carbon sugars again. Okay. If you start a drawing and you make mistakes, really don't worry about it. Drawing is going to help you um, know these molecules and the structure of these molecules even better as you draw them. It's like practice, okay? And that's essentially the best way to study is by practicing, all right? All right, so now we need our phosphates, and we're going to have 1, 2, 3 phosphates on this end, and that's going to be our 5 prime end. And we'll have a phosphate bound to each sugar molecule, like we did on the other side. And again, these are phosphodiester bonds. All right. Now down here, all right, I used black for that. Here is our 3 prime OH group. And this is our 5 prime and 3 phosphates. Okay. So you'll notice the synthesis of the DNA of 5 prime to 3 prime is now going in the opposite direction. Okay, and that's what gives it its anti-parallel status. All right. So now the only thing we need to do is draw the rest of the bases. And here we have adenine. And one thing you'll notice is that the letters I'm putting in are a different orientation than the bases on the opposing strand. And look, even the sugar molecules are in opposite direction as well. Okay, here's guanine. All right, we'll have our blue for our thymine. And then finally our cytosine. Okay, now all we have to do is put in our our bonds between the adenine and the thymine. These are our hydrogen bonds. There are 3 of them, bonding the guanine to the cytosines, 2 adenine to thymine. Okay, so you can see how the geometry of these molecules fit together. All right, um, and that's if you had, say, like an adenine trying to bond, make a base pair between adenine and, say, cytosine, you wouldn't have a complete fit. Okay, there would be some hydrogen bonds formed, but they wouldn't be as strong. Okay, um, as with adenine with thymine and cytosine to guanine. Okay, so this gives us our anti-parallel structure of DNA. All right. Now, there's one other distinction between all of the nitrogenous bases. They fall into 2 categories. And there's a simple mnemonic device that will help you remember which ones are which. Okay. And the simple mnemonic device is going to be all girls. Okay. Let's try blue are pure. Okay. And when they get married, how many rings do they get? That's right, 2 rings, engagement ring and the wedding ring. That's going to note the difference between, okay, here will be adenine, okay, and guanine also has 2 rings. Now you can understand why it's all girls, all right? And they're pure because these are classified as purines, okay? And that means the 2 that you have left, cytosine and thymine, they only have 1 ring between the 2. So here's cytosine. And then thymine. I'm going to do that one in blue. Okay. These are known as pyrimidines. And there you have the whole structure of DNA. And now, what I'm going to talk about is something that's really, really wild. Okay? Um, how DNA is able to replicate itself. Let's flip that over like this. Okay, let's start out with a cell. I'm getting a little warning light that my battery charge is getting a little low. Okay, so there we go. Save that. All right, let's draw a human cell. And I'm not going to try to make this um, um, so exact or so precise. All right, just going to do my best. Okay, most cells in our body, all right, um, have a diameter of about 1.5 micrometers or microns. Um, the unit is the Greek letter mu, all right, and then the letter m, okay, 1.5 microns. The nucleus of the cell only takes up 10% of the volume of the entire cell. I'm going to make that a little bit bigger so we can see the number 46 inside of it. All right, here's our nucleus. And what do you think the 46 refers to? Well, if you thought about these are the chromosomes, you're correct. 46 chromosomes. 23 comes from one parent, and another 23 comes from the other parent. These chromosomes have 3.2 billion base pairs. That's those C to G's, all right, A to T's. 3.2 billion. Notice I'm putting the hydrogen bonds in there as well. Okay. Well, of these 3.2 billion base pairs, um, it codes for 25,000 genes. Okay. So on your drawing, you've got a DNA molecule. All right. I guess I could do the same thing, but um, I'm going to be having my chromosome a little bit further down. All right. If that's okay. Yes, it's okay. All right. So um, if you were to take all 46 chromosomes and unwind all of them and put them end to end, it would take up a length of 6 feet or 1.8 meters in length. Okay. 6 feet of genetic information, right? 1.8 meters. Okay. Let me give you an idea of, how that all adds up. Okay. If we had those 46 chromosomes, okay, let's say this is your textbook, all right? that keeps all, all of the information, your genetic information in. Okay? So here's like my book, my DNA book. Okay? And we'd have all these pages here. All right? And every page that's inside of here is the code, okay, has all of the chromosomes. And this would be like chromosome number one. That's kind of like chapter one, okay? In order for you um, to take up all the pages and copy all of these pages, page two, page three, page four, and these would be the chromosomes, all right? And not just a single chromosome, because you get one from one parent and one from the other parent, okay? So we have 23 pairs or a grand total of 46, all right? um, you would need to type 60 words per minute, okay? um, at 8 hours per day for 50 years to copy all the genetic information. Okay? Now that's kind of puzzling, all right? Because if you recall the cell cycle, oh, I need a new color. I'm going to pick purple. Okay? Got to splash in a little color here. Okay? And I think I put a diagram of that in your notes. Okay? So there is some fill in the blanks. One of the bulleted statements. So we have 46 chromosomes. That's equivalent to 3.2 billion base pairs of DNA. Okay? So if you took all the chromosomes, unwound them, and then attached them end to end, it would measure 6 feet or about 2 meters, or more precise, 1.88 meters. Okay? So remember, and I guess you can go ahead and flip to the next page. All right? We have our cell cycle. Okay? And remember in our cell cycle, we would have like our M phase, that's right here, and then cytokinesis, right here. We would have our G1 phase, right here, and then we would have our S phase, and then G2 phase. Okay? And I think, remember, here is the G sub O phase. This is when cell division is arrested. Okay? um, but remember that the length of time for this to cycle around, completely around, from start to finish, is 22 hours. Okay? Well, that's how long it takes for the entire cell cycle. There is only 1 hour available to actually conduct DNA replication, not 50 years. All right. So, can you imagine trying to copy all of this information, okay, in such a short period of time? It doesn't really seem possible that that can happen. There, we have to figure out how this does happen, okay? So, let me give you an idea, all right? We have, I'm going to draw my chromosomes here, okay? And here's where the centromere is located. And this is what I refer to as being the chromosomes placed at metacentric and metacentric refers to the midpoint of the chromosome. Okay, and here we would have our P arm, and this is our Q arm. Okay. So inside of this molecule, I'm just going to throw in a splash of red. And this is going to be chromosome number 1. Since this has most of the information in here, there are 249 million base pairs. Okay, that constitutes 8% of the entire genome. Okay, all right. So if we were to unwind, all right, uh, um, this chromosome, and this would measure out, unwound, oops, unwind, all right, this would measure 8.5 centimeters. Okay, and here's the thing to realize, you got 8.5 centimeters, okay, just for chromosome number 1. Okay, so here's a ruler right here. Okay, all right, 8.5 centimeters. Now, this is in millimeters. Okay, so here's 1, 2, 3, all the way up to here. Okay, that's 8.5 centimeters here. Huh, this is another puzzle because this is only one, the cell is only 1.5 microns in size. All right, and the nucleus, where all the DNA, all the chromosomes are, only takes up 10% of that. Okay, so you're looking at about 0.15 microns. How is that even possible? Well, all right, that's why I gave you that image of how DNA is highly coiled, all right, by the process of those histones that really condense the genetic material. Okay, and we can just fill this in here. All right, And that's where the DNA molecule is located. It's okay, all right. So here we have a problem. We have this enormous, um, we have 46 or 23 pairs, and this is just chromosome number one, where we have one pair. Okay, now we're going to have 22 more pairs. All right, what we have to do is we've got to unravel all of that DNA. Okay, and replicate it within 60 minutes. How is that even possible? Okay. Well, let's try. All right. You'll notice below the chromosomes. All right. I have a DNA sequence, and it starts off. Here's our 5' end, and here is our 3' end. Okay. And let me put the backbone on here before I'm going to put my arrow in 5 to 3'. And we have adenine, adenine. I'm not going to color code this. And then 2 thymines, 2 guanines, and then 2 cytosines. And for my opposing strand, 5' to 3', we'll have the complementary sequence. Okay. And this is going to be, I'm not going to draw them upside down, but A always pairs with T. So A with T, T with A. Okay, I wonder if you can see that. All right, T with A again. G pairs with C, and C, and G, and G. Okay, now we can put our hydrogen bonds in here. Okay, to this top one, I'm going to label this. This is our template strand. Okay, there's a little call out box that you see on your guided notes. All right, so this one is our template strand. And coincidentally, the opposing strand is our complementary strand. Okay. Now, this molecule would be like the double helix, okay, all twisted in a spiral or a double helix form. It's flat like this just so you can see it, it clearly. All right. Um, now, um, below this, all right, we got a special type. I know this is um, in yellow, but I'm going to do it in orange. There is a fantastic type of molecule that's called DNA helicase. Okay. And yep, he has some buddies. Inside of the cell, there are millions of these enzymes called DNA helicase. And what do you think the name actually refers to? DNA helicase. Remember, it's all in the shape of a double helix. So, what do you think helicase does? Well, if you're unsure, what if I said, well, it does the opposite? Maybe you could figure it out if I said the word anti-helicase. Okay? Okay? If you're thinking about how it unwinds the molecule, you would be right. Okay? I wish I had kind of like a piece of rope, all right, and that you can see all of the strands of the rope, and they're all twisted around each other, all right. And there's only one way of how you can unwind this molecule, and that is now we don't want to start at the ends of the molecule, okay? Because um, um, that's not going to help out in terms of replication. We have to do it in different places along the molecule, okay? So here's what DNA helicase does. All right, it has two functions. It is going to draw it up here. Nip. All right, it's got little teeth. He's going to go up here and break the phosphodiester bond. Okay, so DNA helicase nicks the backbone. And once that you do that, okay, that begins to unwind the molecule. All right. The next thing, with all of his buddies, is going to break the hydrogen bonds. Hydrogen bonds. Okay. Maybe you can't read that too well. All right. So do this one in black. Nix the backbone. Okay. That really, this unwinds, causing the helix to unwind. All right. We'll break the hydrogen bonds. Okay. This is going to unzip the molecule. Okay. So now if I can continue on my last page here, all right. What this does, it creates what's known as replication origins. Okay. So I'm just going to write this up here, and these are actually called replication bubbles. So instead of just having one student like type the entire message, let's get ourselves a lot more typewriters, okay? And a lot more students involved in replicating all of this genetic information. Now you have somewhat of an idea of how much faster this actually can take. So remember, there are 46 chromosomes. I'm really doing a number here as I'm slowing down. For 46 chromosomes, you're going to need 50,000 replication bubbles or origins. Okay, all right. And I guess I can make one of these strands the red. Here we have our template strand. And here we have our complementary strand. Okay, all right. And these are called origins. Okay, and these are called bubbles. Okay, now they're really, I want to put this to bed. Okay, there are no replication forks. Okay, no replication forks. Where did they get the idea of the replication fork? Well, they're just doing it to save space in the booklet. Okay, here's where the replication fork actually is. Okay, it really doesn't exist though, but you will see in some drawings, and mostly if you look online for DNA replication, you're going to see these replication forks. They really don't exist. Okay, if we look at an origin and this large bubble here, there's actually two forks that are joined together. Okay, all right, and here would be two forks. All right, so if you think about 46 chromosomes to make copies and 50,000 replication origins, that's how you can speed up the process of replication, get everything done within 60 minutes. Isn't that special? Okay. Well, there are different types of molecules um, um, that that we need, and we'll call them replicator molecules. Most of them are enzymes. Okay, and we'll start off with DNA helicase. Okay, all right. The next thing, I have like a cursive R and a cursive P. This is an RNA primer. You really can't build a new strand on nothing. You need something to build on. So there is an enzyme that actually builds a single-stranded, okay, molecule. It's actually RNA. Okay, don't worry, it's going to get removed. All right, but we need to start building. Okay, these usually are around 300 base pairs in length. Okay, but I'm not going to draw 300 base pairs on here. I'm just going to represent it as like a little dash. Okay, all right. Remember, DNA helicase, all right, has two functions. It unwinds the molecule, unwinds, unwinds the DNA double helix, and it unzips it. Whoops! Unzips the two strands. Whoa. Sorry about that. A lot of misspellings there. Our next a molecule is an enzyme. Okay. And this is a pretty superpower type of. I made it kind of like, I don't know if you can make this out. It's like a superhero stickman. Okay. This is known as DNA polymerase. And it has a few functions. All right. DNA polymerase adds new bases in the 5' to 3' direction. It can only read it one way, 5 to 3'. Okay? It also removes RNA primers and is a spell checker. So after it builds a new strand, it will go back and check the spelling. Are the A's paired with T's, C's with G's, and so on. Okay? All right? And then our last one is called DNA ligase. And the word ligase is to bind strands together. And more specifically, um, it binds Okazaki fragments together. Okay? So here's where I'm going to make a drawing showing how replication takes place. All right? Make a really big replication bubble. Okay? This is going to be our 5' end with our 3 phosphates. Okay? And this will be our 3' OH. Okay? End here. All right? Now, I'll have the other strand. Let's see. Okay? Whereas over here will be the 5' end with our 3 phosphates. And this will be the 3' OH on this end. Okay? So, let's go ahead and put in our nitrogenous bases. Okay? And I'll try to stick with the same type of color. All right? Let's say we'll have, let me do it this way. Okay? I'll have thymine, adenine, guanine, cytosine, guanine, thymine, adenine, and adenine. Okay? Okay. So remember, we would have a 5 prime going here. Oh, I'm sorry. Um, our 5 prime is here. So this is 5 prime. And here is our 3 prime OH group. Okay. So the first thing is, in order to get this started, all right, is we need to have a primer. And that's going to be right before where we're going to start um, adding the new strand. So here is our RNA primer. It's a short sequence. Sorry about the OH that got in in the way. Okay, and now we're going to have our superhero stickman. Dun, dun, dun. Okay, he's going to bring in the new nitrogenous bases. Okay, and remember, he only goes in one direction, 5 prime to 3 prime. So this new strand that I'm going to be making is going to be um, in a different color. So this is thymine. Oh gosh, you can see it here. This is the thymine. I would have adenine. Okay, it's anti-parallel. Okay, and then I would have thymine, cytosine, guanine, guanine. Okay, cytosine, adenine, thymine, thymine. Okay, now we'll have our strand right here. Okay, and this is where the 5 prime end is. Okay, here's where the 3 prime end is. All right, if you notice, the direction we're going in is 5 to 3 prime. And this is known as the leading strand. Okay? And it's continuous. As DNA helicase breaks more double bond oxygen, more hydrogen bonds, all right, we can keep on adding new nitrogenous bases to our strand. So the leading strand, it's continuous replication. Okay? Now, things are going to happen a little bit differently on the opposing side. So, on the opposing side, all right, is our, remember, this would be our template strand, and this is our complementary strand. Okay, we have our leading strand over here. Okay, so now in black, all right, I'm going to be making the sequence, all right, that pairs with this. And I guess I can do it here. And I want you to try to notice something. Okay, so I'll have thymine, thymine, okay, adenine, cytosine, guanine, guanine, and then, oops, we got a miscue here. Um, oh, no, I don't. Okay, guanine, guanine, and then cytosine, thymine, and finally adenine. Okay. If you notice this template strand, okay, um, this is going to be here. Here is the sequence. All right. A T C G G C A T T. Okay. Let me read the same. Do you notice the same sequence down here in red? A A T G C C G A T. Okay. Let's see. A T. Oh. Oh. Okay. Yeah. Nope. I got it right. Got it right. Oh. Okay. I'm sorry. So this template strand has been reintroduced here in this leading strand. They have the same sequence. T T A C G G C T A. Okay? So now the problem here is with polymerase. All right? Remember, this is our 3' end here and our 5' end here. Okay? Polymerase cannot go in this direction. Okay? Because this direction is 3' to 5'. So, small fragments called Okazaki fragments are going to be produced. Okay, so let's make an Okazaki fragment here, and this will be A Okay, and then this is the 5' to 3' end here. Okay, and then we would have another triplet. All right, G C C, and then our last triplet, G A T. Okay, so now we have these Okazaki fragments that are 5' to 3'. And notice there's a gap. Okay, I wanted to stop and mention, you know, um, it was proposed by a team of scientists, husband and wife. Okay, the Okazaki couple, and Chris Okazaki passed away, I think, in the late 60s, early 70s. But his wife, all right, Dr. Saniko Okazaki, okay, she's 87 years old, and let's give her some hair here, all right. Being a woman, I'm sorry that this is not more not nice looking. I'm trying. I'm trying. Okay. All right. It goes to show, how very few women that there were in science around the 1940s, the 1950s, the 1960s, and 1970s. So it's really, really special that she survived and she became a full professor at Washington University in St. Louis. Um, I really don't know where she's living now. Maybe you can Google her name and find out. Um, it probably would still give her a real boost, if we were to write maybe some letters to her saying, oh gosh, we're studying DNA replication, and we're noticing, the use of your fragments in making what's known as the lagging strand. Okay, so these are going to become the lagging strand. Now we got to do one more thing, and I'm sorry I chose red. Okay, I'm going to change this to purple, this DNA ligase. So what the ligase would do is bind these fragments together. Okay, and remember, we're going from the 5' to 3' in this direction. Okay, so this is known as the lagging strand. And because it's being made in fragments, it's discontinuous replication. Okay, and we would have now on this side of the replication origin. Now you can see, where the fork is. All right, um, and helicase would be unwinding it. You know, it would be unwinding in this direction. Okay, as helicase is actually unwinding and unzipping the molecule in this direction. Okay, so recall that we're going 5' to 3', and we can lay our primer here, okay, and keep, you know, keep adding, all right, making this like a giant leading strand, okay, and pretty soon these origins would actually meet up and have to be the new strands that are getting developed would have to be ligated together. Okay, so one thing that you'll notice is the sequence of this lagging strand that's here in red is going to be the same sequence as our complementary strand. Okay, and the leading strand, all right, is the same sequence as the template strand. Okay, that's an awesome takeaway right there. All right, so you can watch this video over and over again. I think you should watch it again at least once while you go through your notes. It will really help your understanding. Okay, now you're all set to finish the homework. This is Mr. Herzon saying, have a great day.